apologies, I was muted. So I was just saying welcome to everyone. Thanks for listening. I hope you had a good reading week. You're back in the post-reading week hellscape that is Carleton University's fall semester. Let's try to stay positive and enjoy ourselves. You know, folks, the year is just streaming right by, and before you know it, you'll be all finished. And really, before you know it, your whole university career will be just a memory. And the funny thing about memories is that oftentimes, even though they're the experience itself can be a little bit unpleasant when you're going through it, usually memories make it seem a lot better. This is one of the cruel ironies of life, is that everything looks better when you remember it. Yes. So anyway, so I don't mean to ramble or anything like that, but it's hopefully things are going well for y'all, and you had a good reading week that gave you what you needed to relax or recuperate or catch up on whatever it was you needed to do. So let's jump into the old topic. Let's lower the bucket into the content well and pull up some slop for everyone to enjoy. This is our talk on drugs and addiction. So we're going to take a bit of a different format today, or not. I guess not a different format. The format is the same. I'm going to be talking to you over the computer like I always do. But what will be different is the topics in the class. So I guess we'll get into it, but we're not going to be doing so much of the genetics side of things today. It's going to be mostly epigenetics that we talk about. This is just part of my overall plan to vary the perspectives on the conditions that we talk about, so as to give you the widest possible scope on the field of genetics, epigenetics, genomics and epigenomics in the world of mental health and neuroscience. So let's start by reminding you all to check out the progress test for assignment 2. So assignment 2 is due in 1 week from today. That means in about 6 days you should start actually trying to think about doing the assignment. But I'm just kidding, of course. The sooner you start, the better, especially if there's website issues or whatever else. But I kind of got thinking about the second assignment and talking to some students who came by through office hours, and I started to worry a little bit about how, in this particular assignment, unlike the first one, the questions are not self-contained. In this assignment, you're asked to generate a gene list by filtering out genes that are significantly upregulated in your data set. And that depends on putting the right filtering criteria into Excel. And if, for whatever reason, you don't do that correctly, you'll put the wrong gene list into Web Gestalt, and then all of the answers you get downstream of that will be wrong in some way. And that could be quite a bad thing for you if if half the questions on your assignment are wrong. What are we supposed to do as marking? These assignments are not meant to ruin your grades. They're actually supposed to do the opposite. So I thought maybe what I could do is put a little quiz up online on the Brightspace, and you can access it here. You just click this under the quiz section or under the assignment section, whichever of the two. It's not, the points don't go anywhere. You won't earn any points by doing this. You can have unlimited attempts, but all it does is it asks you your answer to a certain question on the quiz or on the assignment. How many genes are upregulated significantly? And it checks to see if you have the right number. And if you have the right number, it very likely means your filtering criteria were set correctly. When you put that into Web Gestalt, you'll get all the answers that you're supposed to, and you can carry on the assignment, and everything will be hunky-dory. If not, it means that there's probably some kind of misunderstanding or mistake. If you continually get the wrong answer, that's a very good sign that you should contact your TA and ask for help on this question, because if you put the wrong number of genes into Web Gestalt, the odds are very good that you will not get the same answers that you're supposed to, and then all of your assignment is going to be really messed up, and nobody wants to see that. So this is sort of a way to pre-check your work and guarantee that you get a good result on your assignment. And then the second question there just asks you to tell us what brain region the study is about. When you go to the biological interpretations question, that's the last question on the assignment, it expects that you understand that there's a certain brain region that this data is from, and we just want to make sure that you know what brain region it is so you don't spout off about something that's irrelevant, just to keep you on track. We're here to help, folks. We're just trying to help. So go ahead and please check that out. I've looked at some students who have done this already, and many of them took several tries to find the right answer. So this is good. I think it means this is working. If those students had put in the first answer they got, they would have had some big problems with their assignment marks. But now that they know, once they arrived at the correct answer, everything should be good. So please take a look at this as soon as you can, and contact your TAs if you need any help. So let's jump into the actual lecture content today. Well, I want to focus on epigenetics for today's class. The reason for this is, like I said, I want to keep things varied, keep things nicely mixed up so that we're not always rehashing the same model of like, okay, here's the GWAS study, here's the genes, here's the whatever, here's the cell types that those genes are expressed in. You kind of, hopefully you'll get that that's a thing that you can do. It doesn't need to be repeated for every single topic. Better, I think, to take the strengths of each topic and use it to highlight a given area of the research. And then once you're out on your own, and if you're curious, you'll probably find that, there's all this stuff I learned about epigenetics in the drug lecture. That actually applies very nicely to, say, stress or to depression or schizophrenia or one of these other kinds of conditions. So I'm trying to build up kind of a mind palace where you can walk around and look through the different windows and take different perspectives on things. So I think the other reason that I've chosen to focus on epigenetics here is that by all, in a truly surprising twist of fate, the genetic contribution to addiction risk actually seems fairly limited, which is strange because I think we're very aware of the fact that addiction seems to run in families. There is heritability on the basis of twin studies. All the signs point to addiction being at least somewhat genetic and heritable, yet when you look at it through the lens of GWAS studies, which is kind of the main type of study we look at, you don't see it. So the GWAS studies, the most up-to-date ones we have, only found a handful of locations in the genome that are linked to addiction. They found 11 for cigarette smoking, 8 for alcohol, and just 2 for illicit drugs, which is quite a surprising thing if you think about it. When you consider how strong addiction can be, it's a little surprising that the genetic apparent heritability of it at the SNP level is as low as it is. So we could go into all of these studies and look at these different things. I mean, part of this also, part of what's also annoying about this is that you're kind of asking a different question for every drug. These studies don't tend to look at addiction as a holistic concept, just because it's too difficult to operationalize. While smoking, while addiction to tobacco, for example, is in many ways the same as addiction to alcohol or to harder drugs, when it comes to the way it affects your life, the effects can be very different. And it just becomes kind of complicated to study it holistically when you're doing genetic studies. So you end up having to kind of break things down by the different kinds of substances that are that you're talking about. And then it just becomes kind of complicated. So we don't really feel like I don't really feel like it's needed to go into the GWAS studies in this time. Let's take a diversion. Part of what fascinates people about the link between drugs and epigenetics is that epigenetics are a type of cellular memory. The big question about drugs that anyone working in addictions is asking is, you take a drug, it has a limited term effect, it affects you for a few hours or whatever length of time it does, and then after a while your body metabolizes it and it's long gone. Yet there are long-term changes that take place in your brain that actually mediate this addiction process. Addiction is, in a sense, it's a type of memory. And it's a type of memory that allows behavioral and neurobiological changes to persist long after the drug is gone from your system. And, we can look at this from sort of a neuroplastic angle about changes in receptor expression and signaling pathways from the midbrain up to the forebrain and all these other kinds of things. But on the level of individual cells, if you're trying to explain how it is that they come to express different genes at different levels for different amounts of time, what you're really asking is about epigenetics. Epigenetics are the type of cellular memory that we would need to explain the neuroplasticity of addiction. So let's look at that a bit more closely. I think to begin, it's worth getting into a discussion about short-term versus long-term epigenetics. And so we haven't, we've talked a fair bit about epigenetics in this course, and my preferred or my favorite type of the epigenetic change, which is DNA methylation. But I've mostly talked about it to you, or described it rather, as a means of maintaining cell differentiation. when your cells are developing and differentiating, they begin in a rather multipotent or pluripotent state where they could differentiate into a lot of different kinds of cells. Cells are characterized by the genes they express. I mean, they're characterized by their shape and location and other things. But one of the main ways that cells are differentiated from one another is just because they express different genes. Liver cells express liver genes, brain cells express brain genes, that kind of thing. And epigenetic mechanisms like DNA methylation and like histone modification are primarily involved in that type of thing, where the epigenetic marks turn on genes that the cells need and try to turn off the genes that the cells don't need. These are patterns that are established early in life, and they tend to remain fairly stable. They can be influenced by things like MQTLs. Recall these methylation quantitative loci. In the case of DNA methylation, can affect the level of methylation at a given CpG on the basis of your genotype at a nearby SNP. Okay, you have that. And you have developmental programming, which can change your epigenetic state on the basis of, for example, how caring and attentive your mother is, or whether you suffered abuse as a child, or whether you suffered from malnutrition, or any of these other kinds of things. So there are variations that can crop up, but for the most part, the type of epigenetics I've told you about is stable and concerned with maintaining cell differentiation. However, this is only the long-term side of the epigenetics puzzle. The short-term side relies on the fact that epigenetic marks such as DNA methylation and histone modification can also change rapidly in response to stimulation. So this is the kind of situation where a short-term exposure to something, for example, a drug, can cause the chromatin or can cause the DNA methylation to be rearranged so as to permanently or in a long-term way change gene expression in a particular cell type. This is the kind of cellular memory that we're talking about. There's also hope, I suppose, that epigenetics could provide a route for treating addiction. What you're kind of thinking about, the kind of riddle you face if you're trying to think about how you would treat drug addiction, is that it is, in essence, a type of memory, a sort of an aberrant, defective kind of memory in the brain, but a memory nevertheless. And if you could somehow target whatever mechanism it is that maintains that memory, you would be able to, in theory, erase it or reverse it or somehow mitigate it so as to, cure the person of their addiction. That would be very nice if we could do that. Now, I've told you before that the epigenome is incredibly complex. This is not good news if you're trying to memorize these things for a test, but it is good news if you're doing research in the field because it means job security. You'll never have to worry about getting laid off because we've solved the epigenome. There's many, many layers to it, and it's unlikely that it's going to be solved anytime soon. So I think I've shown you this figure before, but maybe I can just walk you through it one more time. Perhaps now that you have the benefit of having done a little bit more work with the genome browser and whatnot. So this is a genome browser view, and on the sort of next to the top row are the RefSeq genes. These are the location of different genes along this chromosome. So you've got, PAX5 and FMRPD1 and SHB and ALDH1B1. You got all these genes that are lined up along the top. We're zoomed out a fair ways here. And then layered on top of all these genes in the genome browser view, you have different chromatin features and epigenetic features. So for example, you have RNA-seq. RNA-seq comes to you as reads. The reads pile up, mostly coming from exons in genes that are expressed in the tissue. So wherever you see there's these little signals, that's where you're getting a lot of RNA-seq data. So that means that the genes nearby are being expressed. So that tells you that. Then you've got a whole bunch of histone signals. You've got the H3K9 or the H3, the histone marks affecting histone H3. Wherever you see a signal here, that is a place where there's a lot of that histone mark. So, for example, you could look at this first mark here, H3K36 trimethylation. You can see that there's a lot of it piled up over this SHB gene in whatever this tissue is. There's not very much of it over here by PAX5. So you can kind of compare and contrast, and then you can look at the different types of histone modification and see, wherever there's a lot of this first signal, H3K36 trimethylation, there's also a lot of these other signals, so they maybe have something to do with each other and so on. You have DNase seq. This gives you information on what parts of the chromatin are open and accessible and which parts are kind of packaged away. Areas in a kind of a similar way. And then you have WGBS, that's DNA methylation, and then you have high C, that's the three-dimensional chromatin structure that we'll learn about in some class. So I'm not, obviously this is not the kind of thing that would have any value to you as a topic to remember. You aren't going to have to memorize this figure or any of the information it contains. But what I want you to appreciate is that for any given gene, there are so many layers of information that stack on top of it that give you its exact genetic and epigenetic context. So if you really want to know what's happening with a given gene in a given cell type, you have to know all of this. You'd have to know the levels of all these different histone modifications, where they cluster in the gene, and in what cell type you're looking, plus DNA methylation from WGBS, plus DNase-seq or ATAC-seq to know about the chromatin accessibility, RNA-seq to know if the gene is being expressed, and so on. So again, my point here is that the epigenome is incredibly complex. And I guess you kind of have to keep this in mind when you're looking at the studies I'm going to show you, because they only really ever focus on one or two things. Whichever ones the lab happens to be an expert at looking at, or whichever ones the lab happens to think is important is what they focus on. But be aware that in any given experiment, there's so many more things that you could be looking at the same time. And to really understand things fully, you would need some sense of what's going on with all of these different measures, and actually, more, to be completely honest. Okay. So let's talk about another little fact. This lecture is going to be a little bit heavy on kind of technical details. It's maybe a bit more like the first few lectures of the class where I was giving you like techniques and information of that sort. Just because I think that it's not really going to do you a lot of good to try to memorize all these epigenetic marks from drug addiction. They're just a little bit too complicated and a little bit too disconnected from what your daily experience would be as somebody who thinks of the brain. But alternate splicing is quite important, and it's worth getting it off our chest now so we can have it as a concept that's available to us. RNA is, messenger RNA, I should say, mature messenger RNA, is made up of the exons of genes. Exons make up a fairly small fraction of the genome, around 1% or less, but they're the only parts of the genome that actually code for proteins. In a gene body, going between the transcription start site and the transcription end site, you have a certain number of exons that contain the protein coding sequence of that gene. And then in between them, you have these huge areas called introns that are not protein coding. They're transcribed, but they're ultimately spliced out of the transcript and removed because they don't contribute to the actual protein structure. So in a way, they're a little bit mysterious because they don't, the protein structure is what we're actually trying to produce when we make an mRNA. What's the point of these introns? But they're in the middle there and they take up space. Now, the exons are spliced together so that you end up with a mature mRNA that's only made up of exons, and you can make the protein and do whatever the protein needs to do. And this splicing process turns out to be rather variable. You can in certain cases, leave out exons. You can have, so in this example, you could have one variant of the gene where you have all 3 exons that are included. That would look like this. You can have a second splice variant where only the first and the third exon are included and the middle exon is skipped. There's a lot of different ways you can do this, but the point is that you can mix and match exons to a certain extent to create different mRNAs out of the same gene. Now, this is pretty important because if you think about it, there's maybe 20,000 some odd genes in the genome, and that might make you think that there's only 20,000 proteins that are possible. But in point of fact, there's actually a good deal more because you can alternately splice the genes and mix and match the different parts of them to create different versions of the protein. Maybe you could have a long version or a short version, a truncated version. You could have a version that doesn't have the main active site of the enzyme and just kind of takes up space. It's a little hard to say what these all are for, but the point is that by allowing for splice variation, you create a great deal more room for variety in the sort of the expression and the function of these genes. And RNA-seq, as it happens, is very good at figuring this stuff out because RNA-seq sequences the actual transcripts. So in an RNA sequencing experiment, you would start, you'd start somewhere and sequence along, and you would get to see where the exons join up. That would be part of the sequencing data. So you would be able to say, oh, well, in half the mRNAs, we have a junction between the first and second exon. That must mean that this is one splice variant. But in the other half of the mRNAs, we have a junction between the first and the third exon. So half the transcripts are this first variant and half the transcripts are this second variant. And you can kind of go in and study the different types of splice variants. And oftentimes what you will find in an experiment is that the expression of a gene is held stable, and it's not, stimulus or whatever you're doing doesn't change the expression, but actually changes the splice variation and causes a given splice variant to become more or less common. And because the splice variants can lead to differences in the actual protein, this can have a very meaningful effect on the cell's, on the cell's function without ever changing the actual level of the transcript. You just change it from being one version of the splice variant, or one splice variant to a different one, where the overall level of the transcript is held constant. So this is valuable to keep in mind in the context of drug addiction, because back in 2014 it was discovered that cocaine exposure, and we all know about cocaine, that classic drug that people are always doing, is able to induce alternate splicing of RNA. Now, when you're doing this kind of research, typically what neuroscientists will do is they'll give the mouse cocaine. It's usually done in mice, obviously, or rats or something. They'll give the mouse or rat cocaine, and then they'll look at a given brain region that they already know is involved in drug addiction. So here, the researchers were giving these mice cocaine, and then looking in the NAC. This is the nucleus accumbens. For those of you playing along at home, the nucleus accumbens is one of the main nuclei in the striatum that's sort of a destination for dopamine signaling coming up from the midbrain. So it's part of the mesocortical limbic dopamine system that is a big part of the reward pathway. So of course, cocaine being a dopaminergic drug that activates this reward pathway is going to do a lot of stuff here in this nucleus accumbens. So suffice it to say that cocaine induces a lot of alternate splicing of RNA in the nucleus accumbens. The authors of this study found only 92 genes that were significantly different in expression. 92 is actually a fair bit. I don't know, it's like only 92 is a good number, but anyway, it's not a huge number compared to the number of alternate splicing events. 92 genes were differentially expressed, either up or down regulated, but then 443 genes were alternately spliced in a way that would actually change the protein coding sequence. So this is a fairly big deal, right? Protein coding sequence is king. If you have a mutation, for example, to a protein coding sequence, to an exon, that is very likely to produce some kind of measurable effect. Exon mutations are comparatively rare and have a comparatively high level of a phenotypic effect. So protein sequence is king, and if you are changing the effective sequence of proteins by alternately splicing the mRNA that codes for that protein, then you can expect fairly large things to be happening in the brain in response to drugs. And it should be pointed out that this would be impossible to detect with older methods like qPCR and microarrays. They never would have seen it. It would have been completely invisible to them. So RNA-seq gives us this new window into the transcriptional effects of cocaine exposure that as we are seeing, can actually affect the protein coding sequence. Now, if you take these differentially spliced genes and put them into a gene ontology search, just like the kind you would do for your second assignment, you just take the list of genes and you throw them into something like WebGestalt or geneontology.org or Gorilla or whatever tool it is you like. The authors found that the differentially spliced genes were all related to things like neuron and synapse function, ion channels. This is the actual diagram of it here. So they have things like membrane-bound vesicles and cytoskeleton and cell junction. Cell junction and synapse are one and the same, pretty much. The cell junction is, the synapse is a type of cell junction, right? So there's a lot of hints, anyway, that cocaine exposure has these effects on neuroplasticity by actually regulating the alternate splicing of neuroplasticity genes, things that have to do with synapses and things along those lines. So I think that's a pretty interesting finding if you think about it. This is a type of change to the molecular biology of the cell that would have been invisible prior to the invention of this modern RNA sequencing technology. And it has a fairly sizable effect on what's going on in this part of the brain. So that's just one little piece of information to have in your back pocket. Again, I'm telling you this not exactly because I want you to memorize that there are 443 protein coding splice variants. That's not what I'm interested in. What I'm interested in is opening your mind to the possibility that this is one of the things that can happen when you're exposed to drugs, or frankly, when you're exposed to anything. Splice variants could be changed. So just keep that in mind as one of the many things that can go on inside of cells that are exposed to stimulation of one sort or another. Let's get into the next technical topic, and this is a little discussion of how ChIP-seq works. So your third assignment, which I'm still working on actually making, it'll be released hopefully in the next couple of weeks, involves ChIP-seq. You're being asked to analyze ChIP-seq data. And this is a kind of the third in the trifecta of classic sequencing techniques in genomics and epigenomics. The first was whole genome sequencing. The second is RNA sequencing, and the third is ChIP-seq. And I guess the fourth, which you've also heard about, is WGBS, whole genome bisulfite-seq. So there's actually 4 different kind of sequencing-based methods that are kind of the center point of this course. So ChIP-seq is the fourth one. And I want to tell you a little bit about this now. In your when you do the assignment, you'll read a little bit more about this, and you'll play with some of the data, and you'll get to see how it works in practice. So you'll get a lot more about this as the course moves forward. But for now, let me just tell you about it in my own words. Recall from one of our earlier lectures that the DNA is not just floating around the nucleus willy-nilly. It's kept in a very organized three-dimensional state by its interaction with various DNA-binding proteins. Particularly, the histone octamers are very essential for keeping DNA organized and in the correct shape. These are protein complexes that the DNA wraps around. These histone octamers, there are 8 different histone proteins bound together. There's 2 H3s, 4 different kinds of H2s, and then 2 different kinds of H4s. That's not really too important to memorize. But there are 8 different histone proteins that are bound together in an octamer. That's a thing with 8 parts is an octamer. And then the DNA is wrapped around it kind of like beads on a string. So the DNA wraps around it, forming these things called nucleosomes. And by organizing the DNA in this way, you can massively compact its physical size just the way you can if you take your headphone cables and wrap them up around one of those plastic doodads. Or when you tie up your shoelaces and you tie up the extra lace into like knots or something. They do that, but they also can be modified in a way to change the way the DNA is actually organized and expressed. So histone modifications are what we're typically curious about when we do ChIP-seq. We know that the histones are there. There's no kind of debate that these are the histones that are present in each of these nucleosomes. And by the way, there's zillions of nucleosomes in the cell. Every nucleosome has about 200 base pairs or so of DNA around it, and then the space between nucleosomes is only several hundred base pairs as well. So given the billions of base pairs in your genome, you've got tons of these nucleosomes. The histone modifications are what we're interested in because the histones themselves have little tails that you can add marks to, acetylation, methylation, that kind of stuff. We've already been over that a little bit. So histone modifications are locus specific. They happen in specific parts of the genome and regulate specific genes. So you don't really want to just assess global histone acetylation, although I've seen people do this. It's a totally useless piece of information. You don't want to know just the overall level of histone acetylation. You want to know the level of histone acetylation, for example, in one particular region, in the promoter region of a gene that you happen to be interested in. So chromatin immunoprecipitation, which is what ChIP stands for, followed by next generation sequencing, that's what the seq stands for, is the best way to gather information about both the location and the amount of a given histone modification. So if you are looking, say, for example, H3K27 trimethylation or whatever modification you're curious about, what ChIP-seq will tell you is where that modification is happening in the genome, and for every location, how much is it being modified? Is it a little bit of methylation, or is it a lot of methylation? Is it a little bit of acetylation, or is it a lot of acetylation? This is what ChIP-seq will tell you. So how does it work? Well, it uses a very common kind of concept in molecular biology, which is the fact that antibodies can be made to bind to all kinds of useful biological targets. Now, you might normally think of antibodies as part of the immune system. What they do in the context of immunity is They are produced by your humoral immune system, and they are essentially made to bind to foreign invaders, bacteria and viruses and other kinds of things. Can be recognized by antibodies. The antibodies will cling to the invader and mark that for targeting by other immune cells, or just physically clump it up and make it so it precipitates out of your sort of precipitates out and becomes inactivated and can be cleaned up by your macrophages or whatever. There's a whole bunch of stuff going on with the immune system. But anyway, antibodies happen to be these proteins that have a high level of affinity for their target and bind to it very readily. So that's great for your immune system, but it can also be used commercially. You can actually produce antibodies on a commercial scale that bind to specific target proteins that you're curious about. So you can buy antibodies, for example, on the website Abcam. Abcam makes antibodies, and you can just pick out a histone modification that you're curious about. For example, anti-H3 K4 trimethylation can be purchased. You just have to pay about $535, and I think might be American, but anyway, you pay a few $100s, and you'll be given 50 micrograms of this wonderful chemical, this wonderful compound rather. It's made in a rabbit. It's a rabbit polyclonal antibody, and its only job in life is that if you mix it in with a bunch of chromatin, it will lock on and stick to anywhere where there's H3K4 trimethylation. Any place where that histone is, the antibody will stick to that histone and stick to it very, very strongly. So this is great for Western blots and immunohistochemistry and many, many other molecular biology techniques, but it's also used in this type of ChIP-seq. So what you do first is you grab your chromatin from the cells you're interested in. You just pull you lyse the cells and you mash them all up to break apart all the cellular components. And then you treat the DNA and the chromatin with formaldehyde. This causes the DNA and whatever proteins that are near the DNA to be kind of temporarily glued together. The histones in the DNA are bound by various fairly weak factors that are okay in the context of a cell's nucleus where everything's carefully protected. But once you've lysed the cell and everything's floating around, the DNA and the proteins can dissociate and you lose that connection that you're interested in. So the formaldehyde glues the DNA and the proteins together and keeps them cross-linked so that they can be processed as one unit. Then you break the chromatin into fragments using sonication. This is literally just a ultrasonic kind of vibration mechanism, although you can use enzymes too. And what you end up with then are these little fragments of DNA that are bound up to histones. So this line here represents the DNA, and this circle represents the histone that you're curious about. And you just have these little fragments of DNA bound up with little histones. You kind of create little fragments. After cross-linking, you can take these fragments of chromatin that are bound to DNA and expose them to the antibody and precipitate them out. Now, the antibody would be immobilized to something, often agarose beads. You want the antibody to be stuck to something solid so that when you pass your cross-linked chromatin through, the antibody grabs on whenever the right kind of histone floats by. It'll grab onto it and hold it in place, while everything that's not attached to that histone flows right through. So ultimately, you're precipitating out, or you're capturing all of the bits of DNA that are bound to the protein or to the histone that you're interested in by the use of this immobilized antibody. The antibody can be immobilized a lot of different ways, but ultimately it doesn't really matter for our purposes. But what you've done is you've captured this, and now you can use it for all kinds of things. So after this immunoprecipitation step, you can take the DNA that was captured, purify it out. The proteins now are no longer useful, the antibody and the histone. You can throw away in the garbage, because what you have is more important than the histone itself. You have the DNA the histone was bound to. So now what you have is the DNA in your cells, where you have all of the DNA in your cells that happened to be associated with this particular histone, or whatever it is you're looking at. So then, now that you've got the DNA, all you have to do is sequence it, and then you'll know where the DNA is in the genome. You'll know exactly where in the genome this histone happened to be bound to. And it's a quantitative measure, so it's a little bit like RNA sequencing. The amount of reads that you get in a given area, in RNA-seq, for example, tells you how much that gene is expressed. The amount of reads you get in a given area in ChIP-seq tells you how strongly or in how many cells that particular relationship exists. So if you have a lot of reads coming in, it means that there's a very strong link between that particular histone modification and a given gene or a given region of the genome. So that's how it works, folks. It's really just that simple. And there's a very kind of basic logic to this that appears over and over again. If you can capture out or purify some part of the DNA that is of interest to you because it's bound to a given protein or bound to something that you're curious about, and then if you sequence that DNA, it will tell you where in the genome that protein was bound to. Let me think if there's an example of this, like an analogy. I don't know. I mean, like, kind of imagine, you've got a giant book or something, and somebody has put a bookmark in one of the random pages. If you tear out the page that has a bookmark and read what the page says, you'll know what part of the book the bookmark was in. This is kind of the same idea, I suppose. Like, if you take out the parts of the DNA that are bound to the histone that you're curious about with this antibody immunoprecipitation technique, then you can read that DNA and you'll know where in the genome that protein happened to have been located. And you can work backwards from that and figure out how much of the protein would have been there and all kinds of other useful pieces of information. So here's just a little overview of how it works. One of the things I want you to understand is that when you're sequencing these bits of DNA that you've purified that had been bound to your protein of interest, here they're talking about P53, which is a tumor suppressor gene, but it could be a histone or it could be a transcription factor, it could be a lot of things. Those sequences are read out by a sequencer, and you get the sequence, and then they're mapped onto the genome to a specific spot in the DNA, and then they pile up. They form these things called peaks. So areas where there's a lot of that protein bound, you'll get a lot of sequencing reads from, and when they're aligned, they form these things called peaks where they all pile up in a given location. And those peaks can be transformed into regions. Typically, the peaks are a little bit irritating to use. They're kind of it's a little bit too much information. What you can really do is just take this and say, okay, so in this whole area, there's evidence of binding, and you get this kind of block. You'll see this in practice when you do the assignment, but you'll see this kind of block that represents where that protein happened to have been bound. And then you can take these and do all kinds of downstream analyses with them. You see what I'm saying? Does that make sense? I promise you're going to understand ChIP-seq very nicely by the time you're finished this course. Let what I told you wash over you when you do the assignment. That will also help you a fair bit. And by the time you're finished, this is going to make perfect sense. I remember ChIP-seq was actually when I first started in the world of neuroscience was the year 2008, and I was just finishing my undergrad and decided I wanted to come back for another year and do some work in the neuroscience world, which fortunately there was a chance for me to do that in my university. And the professor I was going to work with told me all about chromatin, which I'd never even heard of up until that point. And at the time, we were thinking of doing ChIP-PCR, which is a very, very early version of this, where instead of taking these bits of DNA and sequencing them, you subject them to qPCR, and you can test one particular region to see if there's more or less of a protein in that one exact area, not the whole genome, just one predetermined area. And then at the same time, I was learning about chip-on-chip, which is when you would take the DNA coming from this experiment and put it on a microarray, not sequencing. So I heard about this many years ago, and it somehow made perfect sense to me, even though I was not a biologist or any kind of expert in molecular biology at the time. So hopefully this will make sense to you at some stage soon as well. The level of detail we have now compared to when I started is just unbelievable how much better things are. So I do have one question. Alexis asks, okay, so peaks are a buildup of information, and these peaks can be transformed into regions. That is precisely it. Yes. So the peaks are just places in the genome where there's a lot of signal coming through, meaning that protein happens to be there happens to be a lot of it in that area. And peaks are kind of a two-dimensional object. You've got a width and a height. That's a little bit cumbersome to work with. It's easier to just transform it into a region. So instead of worrying about it, you set kind of a threshold or a criteria for what defines a peak, and then you transform it into a region. And regions are just things like, chromosome 1, 1000 to 1100, that kind of thing. It just defines a block of the genome. You folks would have worked with blocks of the genome in the first assignment. You just have coordinates, and that's what you end up with. So here is a plot that you're probably not going to enjoy very much, but I'll show it to you anyway. And I don't want to I don't want to stress you too much out about what this plot actually says, because I think that it's impossible to really know, or it's unrealistic to expect that you'll ever need to know this off by heart or what it says. But what I'd like you to know is what it means, because what it means is a lot more important. If you recall back to the slide I showed you earlier, for any given part of the genome, there are dozens upon dozens of different things you can know about. Different kinds of histone marks, different kinds of other epigenetic signals. How do you put it all together? It's kind of too complicated, really, when you add everything up to be able to describe it in a kind of sequential manner, where you say, like, okay, well, so areas that were affected by cocaine had increases in H3K4 methylation, and then decreases in H3K4 trimethylation. And then also, like, there's too many things going on at once to even be able to really describe in words. So the challenge from an informational perspective is, how do you take this huge amount of data and condense it down to like a concept that's actually a little bit more tractable? That's what they're trying to do here. This is Feng et al. From 2014. The same paper we were talking about earlier, actually. What they're trying to do is take all of these signals. These are the histone marks they measured. So they measured H3K4ME1, H3K4ME3, H3K9ME2, and so on and so forth down the line. Also total polymerase 2, that's just RNA polymerase. That's just genes where they're binding. Plus they also looked at RNA-seq, and then they measured whether things were increased or decreased in terms of their level of expression. So tons of information. There's no way to really make sense of that. I don't think it's too hard for my brain to figure out, but maybe you can ask the computer to help you out a little bit. So the authors use what's called k-means clustering. Don't need to worry too much about that, but essentially what they're trying to do are find groups of histone marks that are behaving in a way, behaving in a coordinated way across the different treatments. So they're looking at clustering gene expression and chromatin state. And the authors ultimately created 29 signature clusters. I think this is a very strange way to do it, so I don't want to fuss over this too much, because again, I think it's just not going to help you at all to try to memorize what the authors did in this exact study. It's a fairly esoteric design, but I think it proves an interesting point about how these data can be analyzed. When you have so much when you have so much data coming in that you could never make sense of it with your own brain, the key is to try to apply computational algorithms, feature reduction algorithms, clustering algorithms to try to break it into bigger, or try to combine things into bigger chunks that you can actually understand a little bit. So you can look at all this random data and you will see kind of a cluster or a chunk of things happening here. These are all things that increase, features that increase after the animal has been exposed to cocaine. And you can see that what you have is increasing is H3K4ME3 upstream intergenic in promoter exons and promoter introns, and a few other features like that in these first few clusters. Very complicated, but there is some level of, you can kind of imagine a little bit how you would deal with that, but I don't know. Ultimately, it's just not aesthetically pleasing to me. But there is an important lesson in here about chromatin state. What the authors are trying to do, to sort of limited success, I think, is combine the effect of multiple histone modifications to make sense of things in a bigger picture. What they're trying to do is find the chromatin state. So rather than waste our time looking at this figure, let's look at the concept of chromatin state and try to learn a lesson from that. So the epigenetic situation is very complicated. As I've said many times, you have all kinds of stuff in the mix there. You've got different types of histone marks, you've got DNA methylation, you've got all that sort of stuff. And so every region of the genome is going to have its whatever the baseline genetics are, where the genes are, where the exons are, where the transcription start sites are, etc. And then on top of that, you'll have all of the epigenetics. And the epigenetics can be combined in a way to produce these chromatin states. So let's just go over this figure here. This comes from the Roadmap Epigenomics Consortium back in 2015. This was a very, very big paper led by Manolis Kellis, trying to characterize some of the baseline epigenetic variation that exists in the genome, or exists in humans, rather. Okay. So Let's look first in this part of the figure. You've got about 5 different histone marks. This is just an example with 5. You could have more than this, but you've got H3K4 trimethylation, you've got H3K4 monomethylation, and so on. So these are just things you can measure in the genome with ChIP-seq. You can just put your cells in and measure the amount of this in all the different regions. Then you've got these cells above in this kind of heat map configuration that are colored by their relative enrichment. So they're colored by how overrepresented they are in a given context. So for example, H3K4 trimethylation is heavily enriched in active transcription start sites. Transcription start sites, of course, are where the gene starts and where the process of gene transcription begins, and the active ones are ones where in that particular cell type, that gene is being expressed. So an active TSS in the brain might be on, say, the V-GLUT gene, vesicular glutamate transporter, because cells that express glutamate or cells that produce glutamate need that gene to be expressed. So you would see an active transcription start site. And this particular mark is heavily enriched there. We can go down the line to look at other states, say, for example, a bivalent or poised transcription start site. There, what you'll see is that this first mark, H3K4 trimethylation, is heavily enriched. But at the same time, in the very same place, you also see a strong enrichment of H3K27 trimethylation. Okay? So all of these different features, all of these different chromatin states, are characterized by different combinations of these marks. No state is the exact same. Flanking active TSS, for example, has the first mark enriched, but it also has strong enrichment of the second mark. This next one here, transcription at gene 5' and 3' end, has strong enrichment of all 3 marks, and so on and so forth. So you can kind of go down the line and see that actually, while the situation with regards to chromatin is quite complicated, certain combinations only appear in certain contexts. So the only place you will ever see, for example, the only place you'll ever see H3K4 trimethylation at the same place as H3K27 trimethylation is at a bivalent or poised TSS. By the way, that just means a transcription start site that is kind of teetering between active and inactive. You can have these situations where they're bivalent, meaning they have both inhibitory and permissive marks, or marks that increase expression and decrease expression happening in the same place. Those are transcription start sites that are going to be tipped one way or the other during cell differentiation. I don't think you need to necessarily, I wouldn't, you don't have to memorize what all these things mean. I think it's a little bit too much to ask. But I just want to say that these are all states that are kind of bigger picture things that are relevant to characterizing the epigenetics. And that's what this heat map part means. It tells you about the unique recipe of different marks that goes into each of these features. Over here, it will tell you what percentage of the overall genome is covered by these features. So you'll see, for example, that active transcription start sites make up only 0.7% of the genome. Enhancers make only 2.8% of the genome. And quiescent DNA, this is DNA that is not being expressed, it's just nothing's really happening there. 67.8% of the genome is quiescent, where there's not any expression, there's really very little happening. So that tells you that most of the genome, of course, is not active. Only a very small proportion of the genome has anything actually going on at it. So this is the essence of chromatin state. And this is how things, like how complicated multidimensional ChIP-seq data sets with many, many different marks are broken down. And you can boil down, if you have results, say, from 5 different ChIP experiments, you can boil the results down into these different combinations of co-occurring marks. I think it's important, by the way, to keep this in mind, given the fact that you have, in this particular case at least, 5 histone modifications, you actually could imagine thousands of different combinations and permutations. Yet, you could have equal amounts of all 5, or you could have, maybe 2 and 4, or you could have all kinds of different combinations. Yet, biologically, only a few are actually observed. So of all the possible combinations you could see, only a handful are really seen in real life. This is, I think, a lovely piece of, biological meaning when you're applying these kind of computational algorithms to complicated data, and then it comes out showing that there's really order to what you're seeing. That's a beautiful thing, I think. The programmers of this analysis did not plan any of this. They simply put in the data and let the data tell them where to look. So biology can be a very beautiful thing, I think, when it follows these rules and has this kind of structure and order to it that you can discover. Okay, so that's good. So keep that in your back pockets, folks. Save that information for later. That's going to be useful to you as we go forward. We will talk about these kinds of chromatin states a little bit. But mostly kind of what I'm doing here is I just want to raise your consciousness. I want to let you know that this is a thing that exists and it's a type of analysis that can be done. And we'll see it come up here and there, but I really would like you to just appreciate that it does exist and think about it that way. Okay, let's get into a question about DNA methylation and cocaine craving. Now, one of the big puzzles of drug addiction is that even after you've been exposed to a drug and quit, you tend to crave the drug for long periods during abstinence. It doesn't actually decay over time. It actually becomes more intense. It tends to incubate or intensify. That's a big riddle because normally you would think that say for example, drug tolerance goes up the more you use the drug, and if you stop using the drug, the tolerance tends to go down. But craving is kind of the opposite of that. The longer you go without the drug, oftentimes the stronger the cravings get. So in 2015, Massart and company published a study looking at DNA methylation in the nucleus accumbens following cocaine self-administration. And they basically trained rats to give themselves cocaine by pressing on a little lever. And what they found was that a variety of genes changed their methylation levels after a 30-day incubation period following the cocaine administration. And without getting into the details of the study, it does point to a role for rapid changes in DNA methylation in the incubation of cocaine craving. So what you can take from this is the fact that in people who have been exposed to drugs and have become addicted, even when they're not exposed to those drugs, even when they've quit and they're trying to go off the drugs, there are ongoing epigenetic changes in the brain that are making it harder and harder to stay off the drug. Their brain is actually kind of conspiring to make them relapse in a sense. Now I want to talk to you about another concept. So that's all that I have to say about that. That's just one slide of information about that slide, that just pointing to the fact that changes in DNA methylation are important. Now let me go back to teaching you about yet another concept, not anything about the drug addiction question in particular, but just about a concept. Oh, I've got a question. Let me go back to the chat window here. So Natalie asks, when you said of all the combinations you could see, only a few of them are seen in real life, are you referring to the 15 chromatin states as in what we see in real life? That's correct, yeah. So if we go back to, let me just pop back to that slide. These squares indicate the amount of signal you get from each of these 5 marks, right? And you'll see that there are a certain handful of places in the genome where the only mark you see is this first one. There's a certain number of places in the genome, about 0.5%, where the only 2 marks you see are the first 2. And then another handful where you see the first 3. And then there's places where you don't really see anything. Then there's places where you see the second, number 2 and 3, and so on. But you don't see any where you see all 4 at the same time. You don't see any where you see the second and the fourth. That is kind of what I'm getting at. Is like there's lots of combinations that could exist of these marks that you don't actually see in practice. So you end up with 15 states that capture almost all of the variation that exists in the genome, and 15 states covers them very nicely. So passive DNA methylation, or DNA demethylation, I should say. So we know how methylation is added. It's added by the de novo methyltransferases, DNMT3A and DNMT3B. They'll come in and they'll add methylation to cytosines in the genome. That's very good. It's very difficult to remove methylation because methyl groups are covalently bound to cytosines. You have 5-methylcytosine. It's a covalent bond. It's very difficult to remove. That's in contrast, I should say, to histone modifications, which are fairly easy to reverse. But it's not truly permanent. One way you can reduce the amount of DNA methylation in a given place is by not maintaining it. So if you recall, you have DNMT1. This is a maintenance methyltransferase, and what it normally does is whenever the DNA is copied by mitosis or meiosis, but mostly mitosis, you end up, in CpGs at least, you end up with a daughter strand that's hemimethylated. It has a methyl group on one strand that was, and then the new strand that's copied from it doesn't have methylation. And so DNMT1 will come along and methylate the opposite strand on this hemimethylated DNA. So, you've got fully methylated CpGs. Methyl groups are on both strands right next to each other. That's the beauty of the CpG palindrome. You split off into 2 daughter strands. This is hemimethylated. The methylation group was copied on, or was carried over on one side and is missing on the other side. And then DNMT1 comes along and copies that methylation to the opposite strand, thus preserving it to the next round of replication. However, if you take DNMT1 and you block it or remove it somehow or make it so it doesn't work, then the methylation level in cells will tend to decay after every round of division. Because after the first division, you have hemimethylated DNA that's only half as methylated. And then after the second round, then that methylation is reduced again, because every strand or every DNA replicate that's copied from this unmethylated strand, that becomes fully unmethylated. And ultimately, you end up with very, very low levels of methylation after just a few rounds of cell division. This is called passive demethylation because the methyl groups aren't actually being removed actively. They're just being allowed to kind of decay. It's a little bit like, if you're in a course that you don't like and you stop handing in assignments, you don't drop the course, but you just kind of passively don't do the work anymore. And your mark, of course, will gradually decline as this goes on. So that's sort of the comparison there. In contrast, you have what's called active demethylation. This is a fairly new concept, I think. It's new is a bit of a strong word, but it's new-ish in the sense that it wasn't around when I first got started 10 years ago or 12 years ago or whatever number of years ago that was. So you can't really remove methyl groups from cytosines directly, but you can chemically convert them to things. So there's an enzyme called TET, 1011 translocase, and what it will do is convert 5-methylcytosine, which is methylated cytosine, to 5-hydroxymethylcytosine, 5-HMC. This 5-hydroxymethylcytosine will go through several other steps, and ultimately that cytosine will be removed and replaced with a fresh one. So the way that you can demethylate is by applying a bunch of chemical changes to the cytosines to such a point as that they become damaged and have to be removed and repaired. Don't need to get into the details very much, but ultimately what we're talking about here is active demethylation. So it is possible to remove methylation from DNA. It wasn't thought to be a very common thing. I guess people figured that DNA methylation was basically permanent, but as it turns out, it can be removed by both active and passive means. Why does this matter? Well, it turns out that active demethylation can play a role in cocaine action. So our friends Fang et al. Came back in 2015, and they studied the role of TET-mediated DNA they should say demethylation. See, once again, I found a mistake in my own slides. So if you already had downloaded the slides, please change this before studying from these notes. But anyway, they found the see, I've thrown myself off here. They looked at the role of TET-mediated active DNA demethylation in cocaine addiction. In a very simple model, they just give the mouse cocaine a whole bunch of times, and what they found was that chronic cocaine exposure led to a reduction in TET1 expression in the mouse nucleus accumbens. This is giving us, or it would imply at least, a reduction in DNA demethylation. And as we saw from a different study, cocaine changes methylation in both increases and decreases. So Massert et al. Found that about half the genes go up in methylation and half the genes go down in methylation. So now we have a mechanism that can explain how they can go down in methylation. Of course, we already know how they can go up. That's with the de novo methyltransferases. So you have, well, I guess what we're kind of building up here is this network of epigenetic changes that can regulate the methylation level of genes in response to cocaine or in response to various conditions of cocaine exposure. And demethylation can regulate alternative splicing. Now, we've already established that alternate splicing is involved in the response to cocaine, and it turns out that the changes in the demethylation that we see in cocaine exposure, or that are induced by cocaine, this 5-hydroxymethylcytosine, which is it's the marker of demethylation. These marks are enriched at exon boundaries. That's what this plot here is showing. So they're showing the amount of 5-hydroxymethylcytosine on the y-axis, and then on the x-axis, they've centered it exactly on an exon boundary. So this is just, you've got one exon here and you've got another exon here. They can be separated by an indefinite amount of intron space, but the point is at the very end of an exon is where the boundary is. The level of 5-hydroxymethylcytosine is heavily increased at these exon boundaries and falls off the further away you get. So by the time you're 400% of the distance away from the exon boundary, there's no particular enrichment of 5-hydroxymethylcytosine. So most of this demethylation is happening then around exon boundaries. And it turns out that these exon boundaries are very important in mediating alternate splicing, because when the DNA is being read off and transcribed, the epigenetic marks at the exon boundaries appear to play a role in determining how the RNA itself is spliced. It's a little bit unclear how, but it is happening. So there is a correlation then with changes in splice variant frequency. Splice variants that have increased after cocaine have larger amounts of this 5-hydroxymethylcytosine, and variants that have decreased have less amounts. So that's what we're seeing in this figure here. So the y-axis is the percent of upregulated splice variants, and the percent of 5-hydroxymethylcytosine at upregulated splice variants, and you can see that it's increased at variants that go up and in down, or decrease when they go down. So it's sort of a positive correlation there. And and again, to recall, 5-hydroxymethylcytosine is DNA that's being actively demethylated by the TET enzymes. And we also have evidence that this regulates gene expression. So we know that there's almost 100 or so genes that are differentially regulated by cocaine exposure. The number kind of varies. And it turns out that genes that have increases in 5-hydroxymethylcytosine, that is to say genes that are being actively demethylated, are more likely to show upregulation following cocaine exposure. So this is kind of consistent with the idea that that these active demethylation changes are helping to regulate gene expression. And it kind of makes sense, right? Because genes that have higher amounts of methylation, 5-methylcytosine, tend to have lower expression. So if you're demethylating the gene and removing that 5-methylcytosine, you'd expect the expression to go up, because methylation usually makes expression lower. So if you remove the methylation, you're going to get more expression. And this is a heat map showing how they calculated this. This is another one of these enrichment plots where the green figure means that, or the green color means that a given set of genes is enriched among those that have high levels of 5-hydroxymethylcytosine. So I feel like that's a little bit of a confusing way to print it. Better to just focus on what I've written here. Genes, well, I can explain it to you, I guess. So genes where the level of 5-hydroxymethylcytosine goes up in the gene body are heavily enriched for genes that that are upregulated in terms of their expression 4 hours after exposure and also 24 hours after exposure. The color of the green represents the fold enrichment, or the odds ratio, and then the number inside is the p-value. So very significant effects here. So let me give you a summary. That's all I have to tell you about today. I personally think that maybe you got the hint from the way I was talking, but I personally think that the story with drug addiction is a little bit complicated, and I think it's rather tough to communicate in any kind of organized way that's going to leave you feeling satisfied that you have a story in your mind. So if you've gotten through this lecture and you're thinking, what on earth can I even say about this? There's splicing, there's histones, there's demethylation. What is actually causing drug addiction? What can we actually point to, and where in the brain is it happening? Well, my friends, I don't have the answer for you there. This is one of the big weaknesses of this topic, is that it's not studied very well in my estimation, and it hasn't really crystallized into any kind of a solid understanding. So I'm not able to give you like a comprehensive or even cohesive understanding of what is going on with drug addiction. All I can really tell you is that it seems like exposure to drugs changes a lot of things in the brain. It changes a lot of epigenetic marks, it changes gene expression, and it changes RNA splicing in a way that would more than adequately account for the neuroplastic changes that we see. And the epigenetic mechanisms could easily account for the long-lasting persistence of it. So we have this understanding that these changes are probably essential for drug addiction or for the maintenance of it. But that's about all I can really tell you, because the story has not, has not been crystallized into something that makes very much sense. So my apologies to you for this. I tried to make this lecture a little bit more about the technical side of things, to just load you up with a few remaining methods and technical concepts that will help kind of guide us through the last little bits of this course. Because like I said, I don't think that I can really make the drug addiction stuff make a lot of sense, because there's just a lot going on, and it hasn't really resulted in any kind of clear picture. The good news is that we're going to move into the trinucleotide repeat disorders, Fragile X and Huntington's, in the next two classes. And those, I think, are fairly easy to understand. So if this was a bit confusing to you, don't worry, there's going to be some treats coming for you in the next few classes. Let me just pop into the chat window here to look at some questions. So Caitlin asks, I'm a bit confused. How is 5-hydroxymethylcytosine enriched in the last slide if you said TET1 is reduced? Doesn't TET1 mediate the change to 5-hydroxymethylcytosine? Yes. So I always love when people notice this. So this is the big the big kind of question when so this is the slide that they're referring to here. So we do see a reduction in TET1 in response to cocaine. This is the kind of thing where the authors basically noticed this first and then went in to look at where the demethylation is happening and found that actually there's more of it than they thought. This probably just speaks to the fact that RNA expression doesn't always tell you about protein function. TET1 is an enzyme that acts enzymatically on the DNA to demethylate it. The amount of mRNA there doesn't necessarily tell you how much of that enzymatic activity is happening. There's all kinds of kind of little exercises you can think of. Like, for example, maybe it's kind of a negative feedback loop where the more demethylation that happens, the more the enzyme is downregulated to help keep things under control. Like, there's a million different ways you can look at it. It's not so much that this is all going to make beautiful sense, at first glance. More often than not, you're talking about kind of a conversation between the amount of mRNA and the amount of protein there is, and the two don't always agree. So, yeah, that's the answer to that. Not really an answer, but that's my interpretation of that. What really matters ultimately is that there is more 5-hydroxymethylcytosine, meaning there's more demethylation. And the same amount of enzyme can do more work, even if there's less of the mRNA coding for it. And so I hope that helps a little bit. It's kind of a cop-out, but I wasn't the author of the paper, so I can't say. And then, Justin writes, there's a lot of work left to be done in this field. Cool. I agree. Very cool. Like I say, if you're doing this work, you'll always have job security. People are never going to stop spending money on trying to figure out these problems, and the problems are going to take a very, very, very long time to solve. So there's no risk of you getting sent home because everything was figured out before you got there. And then Alexis asks, let's say you were to make a midterm question based off this lecture. What topics might you focus on from this slide deck? See, this is the kind of question that you should be asking, because I'm kind of I haven't really honestly made, the midterm is still a few weeks away. I haven't made the midterm yet. So it's a little tough to say what I would ask. I think, here we're talking about bigger concepts. So I might ask you something about, compare and contrast splice variation with differences in expression, right? Expression differences and splice variations are two separate things, but they both affect how the RNA actually functions. You can change the protein that's being coded for by splice variants, while the overall amount of the RNA is the same. That can have pretty big effects, even though it would look like expression is being held at the same levels. You might get a question like that. I could ask you about, explain how ChIP-seq works, that kind of thing. Or maybe I could ask I don't know, I think I'd hesitate to I don't want to say what I won't ask, but certain things I think, like if I can't explain them really quickly in class, I wouldn't want to necessarily ask the students that either, because I just think it's a little unfair to like ask students to do things that I can't even do very well. So certain things that I don't think I did a great job of explaining, I'm not likely to ask about. And I think, there's a lot of processes here, like demethylation. I think there's a lot you could ask about that, right? I mean, compare and contrast passive and active. Coming up with exam questions is a lot like filling in blanks. Like if you start with compare and contrast, that's a common way exam questions can start. Once you start from that point, you can think, okay, what would a professor ask us to compare and contrast? Active versus passive demethylation, this type of stuff. So hopefully that will be somewhat explanatory. It's like I've said a number of times, figuring out exam questions or predicting what to study is a lot about understanding your instructor's psychology and the way they tend to phrase questions. So if you give yourself like a few lead-off points, like compare and contrast, or what's the difference between this and that, or what is the role of this in that, if you just make those kind of placeholder questions and then go in, look through the slides of like, okay, what could he possibly ask us to compare and contrast here? You kind of get some ideas. So I don't want to give too much more away, but hopefully that will be a little bit helpful to you. Anyways, my friends, thanks for coming by and participating. We'll leave it there for today, and we'll be back on Wednesday with a talk on, I believe, Huntington's disease, and I think that's going to be a pretty fun lecture. Very different in flavor from this one today, which I think will be hopefully welcome to you all. So have a good rest of your week, my friends, and we'll catch you next time.